Welcome again to Kim's Bioproduction Academy. Uh, from this section now today, we want to discuss about Kcse 2024 Biology, some marking scheme. When I use the next scheme to check of how the papers were marked, maybe the mistakes that our students were making. On now this section, this is paper 1, it's an 8-mark question, and being an 8-mark paper, this paper will be divided to 3 sections. That's section A, section B, and section C. On my first slide, we will first deal with the section A. Then we separate section B, which is the graph, and we separate again section C, which is our essay sections. So from this paper, section A will be carrying on how many parts? There will be 5. Each and every section will be 8 marks. Let's now start to understand about what our students did and how they fared on the exam. So section A we are speaking about is 40 marks, and you can check the first question. You answer all the questions in this section. The following wordy question represents a biological process that occur in green plants. That was the question that they were given. In each and every biology exam, you'll be given a question. You have carbon dioxide plus water. We get glucose plus product E. So the first question that they were being asked is name the organelle where the illustrated process takes place in green plants. Where does this process take place? And where it will be taking place is the chloroplast. You can check it's the chloroplast. Chloroplasts was still accepted, but biologically it's wrong. It was accepted chloroplast, but the question was asking what? Organelle. It was asking about organelle. It was asking about organelle. So it was supposed to be only. It was supposed to be chloroplast. That was the one to be accepted. So number 2, give the likely product E. So the question of photosynthesis is always carbon dioxide plus water, then you get glucose plus oxygen gas. So oxygen gas is what is accepted, but also it's the correct answer, but what also the chemical question was still accepted. That is why you could get your mark. Then Roman 2, suggest ways in which the rate of product E can be increased. We are speaking about how can we increase what? Oxygen released on the process. And the accepted answers which were correct is optimum temperature. Also the account, they accepted, increase in temperature, but it's not a very good answer. The correct answer is optimum. So it means the marking was very fair. Then optimum light intensity, also accepted, increase in the light intensity. Then again, you increase the carbon dioxide concentration. How can we increase the carbon dioxide? We increase by adding the water with. We increase the carbon dioxide by adding. If it was an aquatic experiment, you add sodium hydrogen carbonate to the water, which will increase the concentration of this carbon dioxide. Then again, there would be increase availability or amount of water. We increase water. You know the role of water. Water in the light stage of photosynthesis will be split to release the hydrogen atoms and the oxygen gas, which will be used in the dark stage. We have Roman C. Roman C. Suggest one possible source of carbon dioxide in the above process if it was artificially conducted in the laboratory. So we have said you had what? Sodium hydrogen carbonate. That was sodium hydrogen carbonate. That is what was being added. Also, the accepted addition of sodium bicarbonate, because it will be releasing what? Carbon dioxide. Then again, any hydrocarbon. Then the next one is explain how the illustrated process is significant in nature. So significance of what? Photosynthesis. And the significance of photosynthesis is a purifier in our ecosystem. So we speak about what? Number 1 being air purification. It will be removing or reducing the concentration of carbon 4 in the atmosphere, which regulates amount of oxygen and also oxygen in the atmosphere. You speak about purification. Carbon 4 oxide is not a good gas. And not it being not a good gas, what will happen? This gas will cause, if it accumulates in the atmosphere, it leads to the greenhouse effect. So it has to be eliminated, and this will be eliminated through photosynthesis by being used as a raw material in the process. Number 2, increase in the concentration of oxygen in the atmosphere will do what? Used it during respiration. Then again, when you speak about provision of oxygen and other organic what? Matters. Then provision of glucose. We're speaking about what? This one, it means it's adding energy to the ecosystem by provision of what? Glucose. We're speaking about oxygen is a raw material in the respiration process. The more the oxygen, the more the respiration. The more the respiration process, the more the yield of energy. Then again, we are speaking about purification by removing what? That toxic carbon dioxide from the atmosphere. That was the end of the 8 mark of question 1. Let's go to question 2. Question what? 2. That was question 2. In an experiment, the number of red blood cells in the samples of blood labeled F and G and 2, we're speaking about what? Was estimated. G and H was estimated. Different concentration of salt solutions were then separately added. We're speaking about what? Salt solution being added. The samples after 1 hour, the number of the red blood cells present in each sample was counted and recorded as shown in the table. Blood sample, you have the concentration of the salt, number of the red blood cells. When you add 1.0%, number of red blood cells remain to be the same. When you reduce the concentration to 0.6, there will be fewer cells being observed. Then when you make the concentration to be 0.4, the number of the cells observed, there were none number of cells being observed. Name the physiological process being investigated in the experiment. This process, if you name it wrongly, the rest of the questions will become 0. So you have to be very keen with the process. So the process here that we are dealing, you can see this one is an hypertonic solution. Then you have a red blood cell which is being used. You can use a blood animal cell or what? Or a plant cell, but today we're using what? So the process should be osmosis. The process should be what? Osmosis. You get your first mark. Then you account, we are accounting for the results made in the blood sample F. Blood sample F. When you check the blood sample F, we are saying the first one was which blood sample? We say the first one was blood sample F, then G and H. So it's the second one. The sample F, which is the second one. You can check it was at 0.6. So what happens? The concentration of cytoplasm, we speak about fewer cells observed, no cell were observed. Then we speak now about what? Account for the blood in sample F. Then at this point, account for the difference in red blood cells in F and G at the end of the experiment. So we're speaking about F. What happens in F? We're speaking about what? F. And we're saying the F in an experiment, red blood cells in 3 samples, F, G, and H. So the F is the first one. F is the first one. And you can check the first one, we have 1.0, and the number of the red blood cells remained to be the same. So it means the concentration of the cytoplasm, which cytoplasm? Red blood cell cytoplasm was equal to that 1.0 salt solution. So it means it's isotonic. 1.0 salt solution, percent salt solution, is isotonic to the red blood cell cytoplasm. You get your first mark. We're speaking about what? Concentration of the red blood cell cytoplasm being equal to 1.0 salt solution. That makes it to be what? Isotonic to the cytoplasm. And because of this one, what happens? There is no net movement of water molecule in and out of the cell. And this is why the number of the red blood cells remain to be the same. Hope you understand this language. We are speaking about, you are comparing red blood cell cytoplasm to the solution given. Our solution given is 1.0% concentration. Our red blood cell is in equal concentration to that solution, and because of this one, this means the solution is as isotonic, and because of the isotonic nature, there is no net movement of water molecule in and out of this red blood cell, and that is why the number of the red blood cell remained to be the same. Let's check the second one. You account the difference in appearance of red blood cell sample F and G at the end of the experiment. We are comparing 1.0 and when it was at 0.6, and we are saying number remained the same here, fewer cells observed. For the fewer cells to be observed, it means some of the red blood cell did what? Immobilized. Immobilized is the bursting of the red blood cell. So what happens here? We're accounting the difference. So we're speaking about what? You account for the difference in the red blood cell sample in F was treated with the same concentration. So we're speaking about what? No, sorry, red blood cell in sample F and G at the end of what? The experiment. We're speaking about red blood cell in sample G. Which is that sample G? Appeared swollen, bulging outwards, since the solution was hypotonic to the cytoplasm and there was net movement of water into the cell by osmosis, while those placed in F assumed the normal shape in the solution was isotonic. We're speaking here, the solution that is 0.6 being hypotonic to the red blood cell cytoplasm. What do we mean here? Our red blood cell cytoplasm was hypertonic to that solution. So being hypertonic, it will exert high osmotic pressure, drawing water through the semi-permeable membrane of that animal cell, that red blood cell, by osmosis, leading to those red blood cells, some of the red blood cells doing what? Immobilizing, that is bursting, and that is why the number did what? Fewer number as compared from the initial number. On the other hand, we have explained it's isotonic, and because of the equal concentration of solution 1.0 and that of the red blood cell cytoplasm, there would be a net movement of that water. That's why it remained to be the same. Then, with the reason, predict the possible observations that would be made if the sample of F was treated with concentration of 2.6. We are speaking, can you give a reason and a prediction of the possible observation that would be made if the sample F was treated with a salt solution of 2.6? We're speaking about F being 1.0. So when you make it to be 2.6, and we said 1.0 was what? Isotonic. So it means when you do what? You exceed from 1.0. We have said 0.6 is isotonic. So it means you are moving from isotonic, 0.6, at 1.0 you are at isotonic. Then now it means 2.6 you will be at hypertonic. So it means that solution becomes hypertonic to the red blood cell cytoplasm. So it means the red blood cell cytoplasm will be will become what? Hypotonic. So because now the solution is the one that exerts the osmotic pressure, it will draw the water from the red blood cell cytoplasm through that semi-permeable cell membrane by the process of osmosis, leading to those cells becoming crenated, and it will shrink. So we are speaking about shrinking by a process called crenation. This was our answer. We are speaking about the number of the red blood cells now would be the same. Understand that well. The only time that red blood cells will change is when they immobilize. That's the burst. It means it will be completely destroyed. But if they shrink, the number remains to be the same. It will remain to be the same. The solution will be hypertonic to the cytoplasm of the red blood cell, water molecules will be drawn from the cell, making those cells to do what? To shrink and appear to be crenated. We are countering for the crenated. So we're saying it's about crenation. But when they crenate, what happens? The number never changes. They'll just become small in size. So that was the end of that D. Let's go to E. State the importance of the process above during what? Urine formation. We're speaking about what? Stating the importance of the process under investigation. So you're speaking about osmosis. Osmosis, like you're being told, can you give the role of osmosis in the urine formation process? So during this process, we're speaking about reabsorption of water along the what? Bloodstream. That's from the kidney tubules. So it means what? We conserve water. Not too much water is lost through urine, because much water will be reabsorbed to the bloodstream. You understand the science behind it. At the distal convoluted tubule, that is whereby the pituitary gland will be secreting the antidiuretic hormone. This antidiuretic hormone will increase the permeability of that distal convoluted tubule and the kidney tubules, and there will be much more reabsorption of water in the bloodstream by the process of osmosis. We are done with that. That was question number 2. We are done with our 16 marks, that is 8-8. Let's go to number 3. It's a genetic question. So students, you should understand this one. Since the year 2000 to date, a genetic question has never missed. So that should be an area of strength. You should put this more strength on this area. So let's check now. A genetic investigation on a certain population established that in human beings, premature baldness is controlled by N, which is a dominant gene. N is located on the Y chromosome. In fact, they have summarized for you. It's located on the Y chromosome. It's just a clear indication, and it's telling you this is a sex-linked gene. So when you're doing your crossing, you should do a sex-linked gene. And you're being told this premature baldness is where? On the Y chromosome. So this is how we do our genetic cross now. You are the first question. You work out what? Work out the phenotypic ratio of the offspring of kappa comprising of a normal, comprising of a what? Normal mother and a premature bald man. The man is what? Premature bald. The female always is normal. Female, even if you will not be told, the female should be normal, because the female is having XX chromosome. So, and this gene for premature baldness is linked to what? Y. So the man is the one that will be controlling your steadiness. So the man can either be premature bald or it can be a normal. And we're speaking about premature baldness. It's not about baldness. Baldness always, most people get it on old age. But when we speak about premature, that's a young person having that what? Bald-headed. So these are the rules that we should follow. We have the first parental phenotype, parental genotype, gametes. We have fusion and F1 generation. But now this one is the simple signs. In the phenotype area, we have the female. This is the symbol of a female. We have a normal mother. Then this is a symbol of the male. We have what? Bald-headed father. It's 5-mark question. Then now, this is the parental genotype, the genotype. We speak about what? Female is XX. That is the what? Sex chromosomes, X and X. Then you have the genetic cross. Then you have now X and Y, but now you raise with N. You are linking the N of the premature baldness, and it's capital N. And it's only linked to the Y chromosomes. We have the gametes. So you just cycle, you just drop those ones. You have these genotypes, they make the gametes. These are gametes. This was a genotype. Genotypes are pair of alleles, but now we separate those alleles to make what? Gametes. Then we cycle them, and the cycle should be complete. There should be no any hanging line like mine here. I have a mistake already. Then we have the fusion lines. Fusion line, the first with the second. This other one with the far end. Then we have this one. into the far end, and we have that. So that is the fusion lines. The F1, and it should be correct for you to be awarded the mark. Then we have now X with the X with the Y, N, then X with X, then X with Y, N. So the marking forms, these are 5 marks. This is the first mark. Because it's 5, we will give you, but now we're asking about phenotypic ratio. So it will be, the 1 mark will be from the phenotypic ratio that you will give us. So the four marks will be first mark, second mark, third mark, and the 4th mark. That is the marking, and it's supposed to be done. So from that point now, we have what? We're speaking and a few hours and further with the premature. That was part of our question, sorry. So the phenotypic ratio. You can check our phenotypic ratio. We have 2 normal daughters and 2 bald-headed men. You can check about this point. You can check these ladies, they're normal. The daughters are normal. Then these 2 sons are premature bald. So it's about 2:2. So that 2:2, it will be 1:1. You simplify that what question. So we are speaking about what? 2 normal daughters, 2 bald-headed men. But now here you have to simplify. 1 normal daughter is to 1 bald-headed son. You get your 50 mark. B, what's the probability that this cub would have a daughter with premature baldness? It's 0. 0, no chances. 0%, not. We reject 0/2 or 0/4. The main reason we are speaking about is 0 because this one, give a reason. We are not asking today a reason, but tomorrow you might be asking a reason. The reason is because this premature baldness is only linked to Y chromosome but not X chromosome. Then C, name 2 sex-linked traits in human associated with the X chromosome. We are saying what? Sex-linked. That is the question. Underline that. Sex-linked. But which are linked to what? X chromosome. We have hemophilia. You have color blindness. We are saying you have what? Hemophilia. You have color blindness. You have what? Hemophilia and you have color blindness. Then again, we have what? Dystrophy. Dystrophy. Or what? Muscular dystrophy. Muscular what? Dystrophy. That is for X chromosome, but it's affecting the male. Again, some students may prefer, because the question was not specific to using a genetic cross. Other students used what? They used a genetic unit. They used a Punnett square. The Punnett square is still accepted. But if the question could be very specific to the genetic cross, this should be wrong. If it was specific to Punnett square, our first cross would be wrong. So we do the same things. I'm seeing we are missing something here. We should have the parental genotype as normal. Parental what? Genotype. And our parental genotype, the male symbol and the female. Then the second one should be parental genotype, which is what? But again, we are speaking about parental phenotype is what? Normal mother, premature bald father. Then now we have the parental genotype that was XX, genetic cross, XY, you link to N. Then you do, these are our gametes. This is the male, this is the female. So these are for the male, X, Y, N, then XX. When you do the Punnett square level, that is X and X, those are your gametes. Those are your alleles. Then X, Y, N, X, 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 Y, N. That is it. 1 normal daughter, 1 premature. But that was 2:2. Then we will be done with that genetic question and you get your all 8 marks. So we are already at 24th mark. We are already at 24th what? Mark. We are saying we are going to 40 marks. Let's continue. This is another question now. This was number 3. It was about an ecology question. That is a for 3 work, topic 2. During an ecological study in a lake ecosystem, a group of students made the following observation on feeding relationship among some of the organisms. We have, this was a very simple, ever given food, because they tell you what it's on, which organism feeds on which organism. Lobster feeding on algae, small fish feeding on lobster, worms and caterpillar. We're speaking about lobster feeds on what? Algae. Small fish feeds on the lobster, worms, and caterpillar. This caterpillar will feed on the algae. Worms will feed on the caterpillar. Large fish on the small fish. Penguins feed on small fish, lobster, and worms. Then from the students' observation, construct a food web for the ecosystem. Construct what? A food web for the ecosystem. 5 marks. That was something which was what? Very simple. You can check. We spoke about the algae here. Algae is the producer. That is, and this organism, when you check well, they are aquatic ecosystem. So it's about the algae, which was feeding on the lobster. This algae again, no, the lobster was feeding on the algae. The algae again was being still fed by caterpillar. So you get a mark, you get a mark. From the lobster was being fed by what? Penguins and the small fish. You get a mark, but half a mark. Then from this point, this is the caterpillar. The caterpillar was being fed by worms and the small fish. You get a mark, you get a mark. Then from this point, can you check? When you are crossing lines, we don't cross directly. Get to understand that there should be a curve. Because when you do this is a line cross, which is not accepted. You curve your line, so it means this what? Small fish is being fed by large fish. Large fish feeds on the small fish. Worms again feed on the small fish. Then this small fish was feeding on the lobster. Then again lobster was being fed by penguins. Then these penguins will be feeding on lobster, small fish, and worms. That is it. That was the end of our 5-mark construction. It was very simple. From the food web, construct in 4A a food chain which penguins is a secondary consumer. We're speaking about what? Penguins as a secondary consumer. This one, secondary. Just a direct. When you check this one, this one producer, primary, secondary consumer. We need what? Penguins as a secondary consumer. So it's just algae, lobster, and penguins. Because when you go to the other way around, it will be a long way. Because this one will be producer, primary, secondary, tertiary. So it's not. So it's just this one: algae, lobster, and penguins. That was it. Then from that point now, our next question. The question is not that clear, but can you check prevent exit of carbon 4? The reduction of penetration of light. Let's check. Or we are speaking about prevents exit of air, carbon dioxide, causes suffocation, dead living organism, prevents entry stroke diffusion of carbon dioxide air, limiting or preventing what? Photosynthesis, prevents entry of diffusion of oxygen air, causing suffocation of living organism, reduces penetration. This is about what? Water. It means what? It was about water pollution, and this water pollution is about oils. In case oils, water tanker does what? Spills oil onto the lake ecosystem. What would happen? It could prevent entry of air, that is carbon dioxide, and cause suffocation of those aquatic organisms, and they will die. Number 2, because of that layer of oil, it could prevent entry of division of carbon dioxide. Still like that, it was the same answer. Or preventing division of oxygen, causing suffocation, or reducing penetration of light. You know about these oils reflects light, and when it reflects light, what will happen? It will limit light penetration, stopping penetration, and there will be no photosynthesis again. It may block reducing penetration of light. It may block what? Those stomatal spaces. Then again, or clogs the respiratory surface, causing suffocation. Then, it is about fish clogging the gill filaments. About plants, it can clog the stomach, and clogging of those respiratory service. From that point now, let's go to another question, which was question number 5. Already we had which mark? 4 by 8, that is 28. So 8 by 4. 8 by 4, that is what? We're speaking 8 marks times 4 questions. That is 32, right? Yes, that is 32. So our last question, which was number 5, the following represent a cell structure. Which cell structure is this one? This one is a cell membrane. And you can check the cell membrane, the bilipid layer, the phospholipid layer, and the proteins of molecule. And within the protein molecule, these are the protein molecule, we have the pore. These are the things that we were speaking about. It gives the semi-permeability membrane. So the first question that you are being asked, identify the structure represented. It's just a cell membrane, or you can call it plasma layer, or you can call it plasma membrane. This is just the same things. State 2 functions of the structure. The cell membrane role is about, we are speaking about semi-permeable. Again, it encloses the cell contents. Protection of the inner cell inclusion organelles. We have many organelles inside the cell membrane. So its role is to enclose those cell contents. Then number 2, I've shown you the semi-permeable structures. This will allow selective movement of materials in and out of the cell. Then suggest 2 ways in which the function of the structure can be impaired. How can we impair the function of this structure? You can check we're saying within those cell membranes, we have the spacings called what? And the spacings was called semi-permeable cell membrane. That's giving its properties. So because of that semi-permeability, its membrane will be membrane bond of protein. So those proteins will be affected by extreme pH, that strong acid or even strong base. Extreme pH will lower the semi-permeability. Then again, extreme temperatures, too low temperatures. Because they are proteins, they can be denatured by enzymes. We said they are proteins, again, they can be affected by pH. So too low temperatures will inactivate the enzymes, and high temperatures will denature the enzymes, and that semi-permeability will be lost. It will be lost. Name the 2 structures found in plant cell but not in animal cell. This is a very simple question. Just the chloroplast will be found in a plant cell as cell wall. Animal plant. These are chloroplast. Cell wall. Animals don't have a cell wall. They have just a cell membrane. Subvacuole. Animals don't have a subvacuole. Then we have the tonoplast. These are membrane that will enclose that what? Fluid. Inside what? The tonoplast. Then we are saying that the tonoplast is just a membrane, right? What is the purpose of staining before observing under light microscope? It's just for what? Clarity during observation and make the structures more distinct. When you speak about clear, again, still it was being accepted more visible or more distinct. More what? Distinct. So that was the end of our section A Kcse exam. Hope you have learned. The paper was very simple. There was nothing which was complicated. The marking was bit friendly. So welcome us again. You can just subscribe so that you can get my video on the section B and the section C of this paper. That is the graph and the essay question. Thank you very much for being with me during this session. Appreciated.